Biology 101, Lecture 9. Hello, students. Today we're going to look at respiration and photosynthesis. What cool processes are these? These are very interesting. We're going to look first at catabolism. So we already remember we looked at metabolism and we discussed it. It has two parts, anabolism, which is building up, and catabolism, which is breaking down. Today, in respiration, we're going to look at the breakdown of organic molecules, which is catabolism, and how it releases energy. So, in respiration, organic molecules will be broken down, and we will release a lot of energy. Fermentation is a partial degradation of sugars that occurs without any oxygen in the environment. So without oxygen, we can still respire. It will be called anaerobic respiration. And what we will get is not the end products that we usually look for, which is carbon dioxide and water, where the process is complete. But we will get some intermediate products, which will still have a lot of energy in their bonds to release. But fermentation is only part. It's a partial degradation, so it's respiration. It does release energy, but not a lot. But when we talk about respiration, we're mostly thinking about aerobic respiration, which means there is oxygen present in the environment, and we consume that along with organic molecules, and what happens is we end up with a lot of energy released, and we store that in ATP, which is, as we keep saying, the currency of life and is used to power all our enzymatic reactions that the body needs. Anaerobic respiration is very similar to aerobic respiration, but it doesn't consume other compounds like oxygen. It consumes others, and we will look at that. So cellular respiration, did you know, includes both aerobic and anaerobic respiration. Generally, whenever we talk about respiration, we're always assuming that it's aerobic. When it's not aerobic, that's when we go and we specify it's anaerobic respiration. But aerobic respiration, even as we know it, or cellular respiration, has two parts. The first part is anaerobic, and the second part is aerobic. And although we can use any kind of fuel, we can use fats and we can use proteins to start out to burn instead of just carbohydrates, the reaction is always written as this, as right here. If you look at it, whoopsie. Whoops. Let me see if I can get this to work. All right. We always have this as a starting point of respiration, which is glucose. We could actually use fats or proteins and put them here instead of glucose. But we don't, because it's a little bit more complex and there would be more steps to it. So we just start out with a simple compound, and that is carbohydrates. And then we add oxygen to it, and we end up with complete degradation, which results in carbon dioxide and water. And those are the lowest energy compounds. And then we also yield ATP and a lot of heat, because it is exergonic. And I'm going to try and show you this video, which will probably help in this.
Turn that bite of food into a chemical that a cell can recognize and use as energy. The first step is altering the food into its component chemical compounds and then getting those molecules into your cells. That process is called digestion. Once inside your cells, the process of turning that bite of food into useful energy by cellular respiration begins. The process of digestion results with carbohydrates and other molecules being removed from the consumed food and transported into the bloodstream. From there, nutrients, like the carbohydrate glucose, will leave the bloodstream through a capillary wall and enter a tissue cell. Once inside the cell, cellular respiration will completely oxidize the glucose molecule, releasing high-energy electrons. The overall goal is to make ATP, a storage form of energy for most cells. Cellular respiration is a four-stage process that begins with glycolysis. Glycolysis literally means splitting sugars, and it is the first step of cellular respiration, occurring in the cytoplasm of the cell. Glycolysis consists of two distinct phases, an energy investment phase and an energy harvesting phase. In the energy investment phase, two ATP molecules transfer energy to the glucose molecule, forming a 6-carbon sugar diphosphate molecule. This molecule splits, and the energy harvesting phase begins. During this phase, the two 3-carbon molecules are converted to pyruvate, and ATP is formed. Glycolysis is a 10-step reaction that involves the activity of multiple enzymes and enzyme assistance. In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain. and the carbon dioxide that is produced as a byproduct is released. The goal of cellular respiration is to transfer the energy from the food that we eat daily into ATP that our bodies can use. This process starts with the eating of a snack or meal and ends with capturing the energy from the complete breakdown of the nutrients into energy and carbon dioxide.
All right, so that actually tells you, gives you a preview of exactly what respiration is. And we are going to look at that step by step in every detail. So we're going to start first of all by looking at the principle of redox. Chemical reactions that transfer electrons between reactants are called oxidation reduction reactions, or for short, redox reactions. In oxidation, a substance loses electrons or is oxidized. In reduction, a substance gains electrons or it's reduced. So when we gain electrons, we're actually reducing the amount of positive charge. So that's why it's called reduction, because an electron is a negative charge. So that may be a little bit confusing, but just remember that in reduction, you're gaining electrons, so that means the positive charge is getting to be less. So the most important thing in a redox reaction is looking for the electrons and the positive charges. So in this reaction, for instance, it's just sodium and chloride that are getting together to form sodium chloride, which is salt. So we have sodium. It is going to become oxidized, which means it will lose an electron and it becomes a sodium ion. And then chloride and chlorine, which is going to become reduced, which means it's going to gain the electron. So it has one extra electron, so it becomes reduced. So that is what I mean by that. The electron donor is called the reducing agent. So in this case, the reducing agent is sodium, and the electron receptor is called the oxidizing agent. In respiration, what we see is the general equation for respiration is starting out with sugar or glucose, which becomes oxidized to carbon dioxide, and with the addition of 6 molecules of oxygen, which become reduced to become 6 molecules of water plus energy. During cellular respiration, the glucose is oxidized, the oxygen is actually reduced. Okay. In cellular respiration, electrons from organic compounds are usually first transferred to NAD+, which is a coenzyme. NAD+ functions as an oxidizing agent because it is an electron acceptor. And when it accepts it, then it becomes NADH, which is the reduced form of NAD+, and that represents the stored energy that is tapped to synthesize ATP. NADH passes the electrons to the electron transport chain, and oxygen pulls electrons down the chain to regenerate ATP. Sort of like this. NAD is considered a shuttle. So you can add it with an enzyme, and you get this, which is NADH, and so that is why it is just considered a shuttle. Electron transport chains, why do we have them? They serve a very useful purpose. They can actually control the reaction, and we can save the energy instead of just releasing it in one go. So for instance, on the left hand side you see an uncontrolled reaction where you have hydrogen and oxygen, and you can have an explosive release, you get water, all the energy is lost and in one go or one time, and you can't use it. But if you could do it in a little tiny step by step or piece by piece, if you could release all this energy little bits at a time, you would actually be able to harvest it and use it in ATP, which can then be used for cellular respiration and for other problems that we have. So when we harvest energy from glucose, there are 3 stages to cellular respiration. The first stage is called glycolysis, and as you can see from the word, lysis means breaks down, and glyco is glucose. So glycolysis will break down glucose into 2 molecules of pyruvate. So please remember, 2 molecules of pyruvate, not 1. The next stage is the citric acid cycle, also known as the Krebs cycle, and that completes the breakdown of glucose. So glucose is changed into pyruvate in glycolysis, and then pyruvate is taken in the Krebs cycle, and it is completely broken down into carbon dioxide and water. And the last part of cellular respiration is the oxidative phosphorylation, which accounts for the ATP synthesis. Okay, so if we look at it in a diagram, this is what we would actually see. So we start from the most left part of the picture, and it's this teal blue color. You see glycolysis. You're taking glucose, you're changing it to pyruvate. This happens in the cytoplasm, and you get this compound. Some ATP is released, but not much. Then the pyruvate is transferred into the mitochondria. Here, the pyruvate is sent into the Krebs cycle, and ATP is produced. And then after the next part is after the Krebs cycle is the oxidative phosphorylation, where the electrons go through an electron transport chain, and you get more ATP released. Okay, so the process that generates most of the ATP, about 90% of the ATP, it is generated in oxidative phosphorylation, the last part of cellular respiration, because it is powered by redox reactions. A smaller amount of ATP is formed in glycolysis, and a little bit is formed in the Krebs cycle by substrate level phosphorylation. But the majority of all the ATP is formed in the very last part, which is oxidative phosphorylation. For each molecule of glucose degraded to carbon dioxide and water by respiration, the cell makes up to 32 molecules of ATP, sometimes even 36. So let's look at glycolysis. Glycolysis breaks down glucose into 2 molecules of pyruvate, and it occurs in the cytoplasm, and it has 2 major phases. The first phase is called the energy investment phase. The second phase is called the energy payoff phase, and we're going to look at these. What interesting terms are these anyway? Glycolysis occurs whether or not oxygen is present in the environment. So it doesn't care. It doesn't care if there's oxygen. It doesn't care if there's no oxygen. It's going to happen anyway. So this is where we are in the beginning of respiration. We're in the cytoplasm. In the blue box, glycolysis is occurring, and a little bit of ATP is being produced, and also pyruvate is coming out of the breakdown of sugar, and it's being exported into the mitochondria for further processing. So in glycolysis, the 2 phases are the energy investment phase, which you see in the top. So you have glucose. You will invest 2 molecules of ATP, and then you'll end up with 2 ADP molecules and 2 phosphate ions. But in the energy payoff phase, you take 4 ADP molecules, 4 phosphate ions, and you form 4 ATPs. Kind of cool, no? Because you invested 2, you form 4, so you still have a net of gain of 2. 2 ATPs is still the gain. And the next stage is you take the NAD shuttle and some electrons and hydrogen ions, and you get NADH, and you end up with pyruvate and water. So at the bottom of the slide, you see all the 2 phases added up. So glucose produces 2 pyruvate and 2 oxygen, two waters. The energy payoff phase is where 4 ATP are formed, 2 ATP are used, and you end up with a net of 2 ATPs. And the last part is 2 NADH plus 4 electrons plus 4 protons, and you end up with 2 NADHs and 2 protons. In the presence of oxygen, pyruvate will enter the mitochondria of eukaryotic cells, where the oxidation of glucose is completed. Before the citric acid cycle can begin, pyruvate has to be converted to acetyl coenzyme A, or acetyl-CoA, which links glycolysis to the Krebs cycle. This step is carried out by a multi-enzyme complex that catalyzes 3 reactions. So we're going to look at it in a diagram first. So you have pyruvate on the very left-hand side in the cytoplasm, and it is being sent into the mitochondria with the help of a transport protein. And it is joined by coenzyme A, and it ends up becoming acetyl coenzyme A. So this cycle will complete the breakdown of pyruvate to carbon dioxide. It oxidizes the pyruvate. It generates 1 ATP, 3 NADH, and 1 FADH2s. So the citric cycle actually has 8 steps. The acetyl group of acetyl coenzyme A joins the cycle by combining with oxaloacetate, and it forms citrate. The next 7 steps decompose the citrate back to oxaloacetate. The NADH and the FADH2 produced by the cycle relay electrons extracted from the food to the electron transport chain. That's all they do. So if we look at a picture of this, we take the pyruvate from glycolysis, and we're going to go and oxidize it. So two molecules of pyruvate per molecule of glucose. We will add acetyl coenzyme A, and we end up with a compound called acetyl coenzyme A, which will enter the citric acid cycle. And as you can see, ATP is produced, and 1 molecule. And NAD plus is reduced to NADH, FAD is changed to FADH2. And if we were to look at all 7 steps, all 8 of them, plus the first one, which we looked at in big detail, this is what would happen. We take oxaloacetate, we add acetyl coenzyme A to it, you get citrate, which will then change into isocitrate, which will then be acted upon by other enzymes to get alpha-ketoglutarate, and then that changes into succinyl coenzyme A, and then into succinate, fumarate, malate, and back to oxaloacetate. So that is why it's called a cycle, because you cycle through all these products, but you end up with oxaloacetate one more time. So following glycolysis and the Krebs cycle, NADH and FADH2 account for most of the energy extracted from food. These 2 electron carriers donate electrons to the electron transport chain, which powers ATP synthesis via oxidative phosphorylation. The pathway of electron transport, how does that work? Well, the electron transport chain is in the inner membrane of the mitochondria, which are called cristae. Most of the chain's components are proteins, which exist in multi-protein complexes. The carriers alternate reduced and oxidized states as they accept and they donate electrons. So they, at one point in time, they're reduced, then they become oxidized, then they become reduced, then they become oxidized, then they become reduced, and then they become oxidized, depending on if they're accepting or donating electrons. And electrons drop in free energy as they go down the chain and are finally passed to oxygen, forming water, like this. So here we have the electron transport chain. We have all the proteins and the cytochromes in it. What we are seeing is that the free energy, the Gibbs free energy relative to oxygen per kilocalories of mole. And you end up with water, oxygen, and we have gone through the electron transport chain, and we're now looking at why did we go in there, because no energy is actually released in the electron transport chain. The electrons are only transferred from NADH or FADH2 to the chain, and they're passed through a number of proteins called cytochromes. Each of them has an iron atom to oxygen. And it does, the important thing to remember is that the electron transport chain does not generate any ATP directly. All it does is it breaks the large free energy drop from food to oxygen into smaller steps that will release energy in manageable amounts. If you remember in the animation we saw, you saw all these little cytochromes moving, passing the electrons, and then what you saw was the production of protons, which went in the intermembrane space, and then it went and formed, went into this complex, which we're going to look at in a moment. So, because of chemiosmosis, the electron transfer in the electron transport chain causes proteins to pump protons from the mitochondrial matrix to the intermembrane space. That's what I had just reminded you, that we saw this in that video. Then we also saw the protons moving back across the membrane, passing through the protein complex, ATP synthase. And ATP synthase uses the exergonic flow of the protons to drive phosphorylation of ATP. This is an example of chemiosmosis, the use of energy in a proton gradient to drive cellular work. And although I'm not going to show you that animation again, but we can see it in one dimension right here. And so we see this is a phospholipid bilayer membrane. Let me see if I can find my pen. Yeah, it's right here. So here's the phospholipid bilayer membrane. Yep, the whole thing. And we have in between these proteins, or these complex of electron carriers. So they're just going to shuttle the electrons from one to the next. So it goes there, it comes back, gets another one, comes back, goes. It keeps on doing this. And this next one will do the same thing to the next. And then what will happen is the protons get expelled into the intermembrane space. Here they float around and they go and join up with ATP synthase, which then goes down and it takes ADP and 1-phosphate and makes ATP from it. So it's awfully cool, because what we're seeing here is everything working together. We see chemiosmosis at the end, and we see the electron transport chain in the beginning, and the combined electron transport chain and chemiosmosis are called oxidative phosphorylation. The energy stored in a proton gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis. And the proton gradient is referred to as a proton motive force, emphasizing its capacity to do work. During cellular respiration, most energy flows in this sequence. You start out with glucose, you take NADH, then you go to the electron transport chain, then you use the proton motive force to produce ATP. About 34% of the energy in a glucose molecule is transferred to ATP during cellular respiration, making about 32 ATP molecules total. there's several reasons why the number of ATP, sometimes people say 30, sometimes it's 36. It's okay. Don't worry about that too much. Just generally think that it makes about 32 ATP. In this diagram, which is sort of like a summary adding up of what the ATP, where the ATP is formed, if you see on the far left-hand side, glycolysis happens. Glucose is changed, one glucose molecule is changed into 2 pyruvate molecules, and you end up with 2, you end up with 2 ATP. And then this pyruvate is shuttled off into the mitochondria, where it is added on to acetyl coenzyme A, and we enter the citric acid cycle, and it just keeps on going around and around and around and around. And then we end up with this going into the oxidative phosphorylation or the electron transport chain. And what you have from the citric acid cycle is just 2 ATP, but in the oxidative phosphorylation you can get either 26 or 28 ATP. So you end up with 2 plus 2 is 4, and 26 or 28, you get either 30 or 32 ATP total. So that is why it varies, but the maximum number per glucose molecule is 32. In anaerobic respiration, there is no oxygen around. Cellular respiration requires oxygen to produce ATP. As we saw in the mitochondria, oxygen is needed. But without oxygen, the electron transport chain can't work. It ceases to operate, so glycolysis couples with anaerobic respiration or fermentation to produce ATP. Anaerobic respiration uses an electron transport chain with a final electron acceptor other than oxygen. Fermentation uses substrate-level phosphorylation instead of an electron transport chain to generate ATP. So there are 2 types of fermentation. Fermentation consists of glycolysis plus reactions that regenerate NAD+, which can be reused by glycolysis. 2 common types of fermentation are alcohol fermentation and lactic acid fermentation. So fermentation by, so we see glucose over here. I'm going to get my pen again. So we take glucose, we do glycolysis, we get 2 pyruvate, 2 ATPs, and we're good. But if there's no oxygen, this pyruvate cannot go there into the mitochondria to go make more ATP. So we could then, therefore, what are we supposed to do? We've got this pyruvate. We could go into this other cycle, because that door is closed. Can't go there. So we go into this other cycle in anaerobic respiration. Yeast is a facultative anaerobe, so it can use oxygen, or if there's none present, it doesn't have to have it. And so at this point, the pyruvate is going to be changed to ethanol and carbon dioxide, not carbon dioxide and water, but ethanol. So there's still some energy left in here that's not utilized. Or if we're not yeast and we're people, then we can use it, use the pyruvate change it into lactate, which again is not the last lowest state. It's not carbon dioxide and water, but it does it does synthesize some ATP. So 0 ATP would be bad, but some is better than none. In alcohol fermentation, pyruvate is converted to ethanol in 2 steps. The first step releases carbon dioxide, and the second step produces ethanol. Alcohol fermentation by yeast is used all the time in brewing, winemaking, baking. And in class I did say that if we did not actually put yeast inside flour, we tried to eat the bread that came out of the oven without the yeast, and we chomped on it, our teeth would break because the bread would be as hard as a rock. So the yeast is a very useful organism to dump into your dough, because those bubbles that it forms of carbon dioxide evaporate, and it leaves the shell of the bubble inside, or the form of it, inside the bread, which makes it fluffy and edible. In lactic acid fermentation, pyruvate is reduced by NADH, forms lactate as an end product, but there is no release of carbon dioxide. So this is not so good because you have a carbon dioxide buildup. Lactic acid fermentation by some fungi and bacteria is used to make cheese and yogurt. So we have used this and capitalized on this idea to make some other kinds of cheese and yogurt are partially degraded milk. So there's still a bunch of energy left in there, and we could actually go and, complete it by eating this stuff. Human muscle cells use lactic acid fermentation to generate ATP when oxygen is scarce. So after you've run a lot and you start getting cramps in your legs, well, that's because of the carbon dioxide buildup and the lactic acid buildup. And until there's, and your body will stop. You won't be able to run anymore because it's too painful until that lactic acid is gone. It is completely degraded into oxygen and carbon dioxide. On the left-hand side here, you see alcohol fermentation, and on the right-hand side, you see lactic acid fermentation, and both of them are taking glucose from glycolysis and then using that pyruvate into making either ethanol or lactate. The differences between fermentation, anaerobic, and aerobic respiration, what are they? Well, what are the similarities first? All of them use glycolysis, and they produce two ATPs they use two ATPs to oxidize glucose and harvest chemical energy of food. In all three, NAD+ is the oxidizing agent that accepts electrons during glycolysis. Cellular respiration produces 32 ATP per glucose molecule, but fermentation only produces 2 ATP per glucose molecule. The differences between all these three: the processes have different mechanisms for oxidizing NADH. In fermentation, an organic molecule, like pyruvate or acetyl aldehyde, acts as the final electron acceptor. In cellular respiration, electrons are transferred to the electron transport chain. So that's a difference. When we talk about aerobic and anaerobic, and we talked about yeast, and I said it was a facultative anaerobe, what does that actually mean? So bacteria can either breathe in oxygen or they can breathe without oxygen. And then there are those who can breathe either in oxygen if it's present, or if it's not present, they'll breathe without it. So the ones that cannot breathe unless there is oxygen are called obligate. But the ones that can go either way, and they can, if there is oxygen present, they'll use it. If there isn't, they'll go into anaerobic respiration. Those are called facultative anaerobes. Obligate anaerobes carry out fermentation or anaerobic respiration, and they cannot survive in the presence of oxygen. But yeast and many bacteria are facultative anaerobes, which means they can switch to either fermentation or cellular respiration. For a facultative anaerobe, pyruvate is the fork in the metabolic road that leads to two alternative catabolic routes, like this. So if you look at the picture at the top, there's glucose. Make it into pyruvate by glycolysis, and now you have to choose, where do you want to go? If there's no oxygen present in an anaerobe, facultative anaerobe, well, it's just going to make ethanol. But if there is oxygen present, it's going to say, well, I'll just shuttle that off to my mitochondria and I'll carry on aerobic respiration. Glycolysis, therefore, is a very ancient process because this is the first part of the degradation of food molecules. Ancient prokaryotes are thought to have used glycolysis long before there was oxygen in the atmosphere to generate ATP. Earth is on its third atmosphere right now. The first atmosphere was pure hydrogen. The second atmosphere was a reducing atmosphere, which means there was ammonia, methane, and other nasty, noxious gases. And at that time, there was no oxygen. Currently, our third atmosphere, we have 79% nitrogen, 21% oxygen, and just tinge of trace gases. But before this present atmosphere, there was no oxygen. So, but there were prokaryotes, and that's why they had to do glycolysis and to live with just the 2 ATP produced by partial degradation. Glycolysis and citric acid cycle are major intersections to various catabolic and anabolic pathways. Glycolysis accepts a wide range of carbohydrates, and proteins are digested to amino acids, and the amino acids either feed glycolysis or the Krebs cycle, and fats are digested to glycerol, which is used in glycolysis, or fatty acids, which is used to generate acetyl coenzyme A. But the difference is an oxidized gram of fat produces more than twice as much ATP as an oxidized gram of carbohydrate. So this is sort of like a summary again. It's always good to have a bunch of pictures around. So on the top of the screen, what you see are different foodstuffs and what happens to them. So usually we talk about carbohydrates being used in respiration because they're changed sugars. Sugars will go into the glycolysis. We always think of glucose, which will then become pyruvate, and that's gone on to other places like Krebs cycle. But we could also have proteins. In fact, we do. When there are no more sugars, then the body has to look for alternate supplies of energy, and it will first look to fats, and then when all the fats are gone, it will look to proteins. That is not preferable. That is actually very non-preferable, because you don't want to break down muscle cells. But the body is, at this point, has no carbohydrates, no fats, no food is coming in, and yet all the respiration and all the processes have to go on, and it needs ATP. So where is it going to get it from? It's got to do it. So it will break down the muscles or the proteins, hoping that one day there will be food and we can regenerate whatever ground we've lost. So the proteins will become amino acids. Ammonia is going to be released, and that product will go into the citric acid cycle so that it can go into oxidative phosphorylation, and we can get a bunch of ATP. So while this is such a complex process, there has to be regulation of it. Otherwise, it would go wild. So feedback inhibition is the most common mechanism for metabolic control, and it's no different for respiration. If the ATP concentration begins to drop, respiration will speed up. When there's plenty of ATP, respiration will slow down. So that's a negative feedback loop. Control of catabolism is based mainly on regulating the activity of enzymes at strategic points in the catabolic pathway, as in here. So we see glycolysis. The inputs are glucose, goes into glycolysis. You get 2 pyruvate plus 2 ATPs, which are the outputs, plus 2 NADHs. In the citric acid cycle, what do you get? You get the inputs are 2 pyruvate, become 2 acetyl coenzyme A, and they go in the citric acid cycle, and you end up with 2 ATPs, 6 carbon dioxide, 2 FADH2s, and 8 NADHs. And then you go into the electron transport chain, where you have the protein complex of electron carriers, and you have NADH, which is carrying the electrons from food, feeding these protein complexes, and becoming reduced into NADH plus. And this goes on, and the protons are released into the intermembrane space, but they're caught up with ATP synthase, which is that giant big purple blob on the side here, and that will then produce the ATP that we so dearly crave. So photosynthesis, however, is the opposite of respiration. And this is just so cool. Although we talk about respiration using up all this energy, photosynthesis does the exact opposite. It produces ATP. Photosynthesis converts solar energy into chemical energy. And there are organisms that will actually do this for us. They're called autotrophs. They sustain themselves without eating anything derived from other organisms. They just take water, carbon dioxide, this is available from the environment, and solar energy. And then they produce organic molecules, which have a lot of solar energy trapped, and we all use that. So you can say that the autotrophs are the producers of the biosphere. They produce organic molecules from carbon dioxide and other inorganic molecules. And photosynthesis occurs in plants, algae, certain other unicellular eukaryotes, and a few prokaryotes. And how does photosynthesis occur? Well, it occurs in the leaves, mostly in the leaves. Because the leaves have chloroplasts, and they are found in the cells of the mesophyll, which is the interior tissue of the leaf. Each mesophyll cell contains about 30 to 40 chloroplasts. Carbon dioxide enters, oxygen exits the leaf through microscopic pores called stomata. So when we look at the structure of a chloroplast, it has an envelope of two membranes surrounding a dense fluid called the stroma. And they look like a stack of pancakes, and they're actually called thylakoids. They're connected sacs in the chloroplast, which compose a third membrane system inside that cell. Kind of cool. Oh, look at all these membranes. Chlorophyll, which is the pigment which gives the leaves the green color, resides in the thylakoid membranes. Thylakoids are stacked in columns called grana, sort of like this. So what we see here is one chloroplast. Inside the chloroplast are these thylakoid membranes. They're stacked like little pancakes. Yeah. And these are the ones that are actually going to harvest the energy for us from the sun. Here is a blow up of it. You see a chloroplast, the thylakoid, this thylakoid space, and at the bottom of the page you actually see a real picture, an electron picture, a micrograph picture of a real chloroplast instead of a model as you see on the top. Photosynthesis is also a series of reactions, and it can also be summarized in one line. And you start out with 6 carbon dioxide molecules, 12 water molecules, plus you add some solar energy, and you end up with, after a bunch of reactions, 1 glucose, 6 oxygen, and 6 water molecules. The overall chemical change during photosynthesis is the reverse of that in cellular respiration. Chloroplasts split water into hydrogen and oxygen, incorporating the electrons of hydrogen into sugar molecules and releasing oxygen as a byproduct. Lucky for us, because we actually need that oxygen to breathe.
Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions. Plants take water, sun, and CO2 to make glucose, the sugar that they use for food. They also put oxygen into the air so we can share, because oxygen is everywhere. The energy transforms from solar to chemical, all the time, everywhere. The process is identical in every plant and every tree, enabling all living things to be. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions and synergy. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions. Let's take a look at the light reaction, making chemical energy when light is captured. Electrons flow through the photosystems, flowing so fast you might have missed them. Within the chloroplast lies the action. The thylakoid membrane is where it happens. ATP and NADPH are products of light, water, air, and space. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions and synergy. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions. The Calvin cycle or dark reaction doesn't need direct sunlight for it to run right. CO2 from the air enters the chloroplast, mixing with organic molecules for the last time with the help of the enzyme Ribisco. Carbon fixation, I thought you knew this, yo. It forms a carbohydrate known as G3P. It's created, recreated, and recycled, you see. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions and synergy. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions. Leaves on trees and even weeds meet needs, making oxygen we need to breathe. You wanna learn it with these, you wanna master the keys. It's photosynthesis, listen to this please. Leaves on trees and even weeds meet needs, making oxygen we need to breathe. You wanna learn it with these, you wanna master the keys. It's photosynthesis, listen to this please. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process of chemical reactions and synergy. Photosynthesis, let's get into this discussion of sunlight and transfer of energy. Photosynthesis, let's get into this process.
Well, that was a very pretty interlude, but it was in the middle of it, there was some very good information. So when we look at it in a boring way, which is like this, we have the reactants, 6 carbon dioxides, 12 waters. With the addition of solar energy, you get these products, which are glucose, water, and oxygen. And the good thing about this picture is that you can actually see where are those atoms going to and from. So when you look at it, this carbon goes into glucose. What happens to the 2 oxygens? So 6 times 2 is 12. So you have 12 oxygens. So 6 of them are going to go here. And the other 6 are going to go here into water. And what happens to these hydrogens? So there's 2 times 12 is 24 hydrogens, yeah? And you have 12 of them over here. So where are the other 12? They're going to go here. 6 times 2 is 12. So that's how you get it to be all balanced out. And then, of course, the last part is the 12 oxygens, and they become 6 molecules of oxygen. So that's just so you know what is going on and where. Photosynthesis is a redox process. It reverses the direction of electron flow compared to respiration. It's a redox process in which water is oxidized and carbon dioxide is reduced. Very neat, isn't it? I mean, you can actually do the reverse. Photosynthesis is an endergonic process. That means it requires energy, which is a good thing because solar energy is provided, and so it can happen. This is the reaction written in redox terms. So carbon dioxide becomes reduced to become glucose. Water becomes oxidized to become oxygen. So different, right? There are two stages of photosynthesis. Photosynthesis consists of the light reactions, which is the photo part of photosynthesis, and the Calvin cycle, which is the synthesis part of photosynthesis. The light reactions occur in the thylakoids. What do? They split water, they release oxygen, they reduce the electron acceptor NADP, not H, NADP plus to NADPH, and they generate ATP from ADP by photophosphorylation, not oxidative phosphorylation. The Calvin cycle, which does not happen in the thylakoids, but it happens in the stroma, forms sugar from carbon dioxide using ATP and NADPH. The Calvin cycle begins with carbon fixation and incorporates carbon dioxide into organic molecules. Whoopsie. Here's a picture of what we just said. So this is supposed to represent one chloroplast. And in this one chloroplast, we have a stack of thylakoid membranes, which are depicted as those green pancake stacks. And that's accepting light from the sun, and it's accepting water from the cell itself. And the reactions occur inside the thylakoid membranes, and you get the expulsion of oxygen. All right. At the same time, ATP is produced by the light reactions, and it is sent into the Calvin cycle, and NADPH goes into the Calvin cycle. The Calvin cycle takes in carbon dioxide, and it produces ADP and phosphate ions and NADPH, and puts it back into the thylakoids to continue their reaction. So the stroma and the thylakoids act in concert. One does one thing, hands it to the other does another thing, hands it back to the first. Okay, and you end up with some sort of sugar after the Calvin cycle. The photosynthetic pigments. So what are those? Those are the substances that absorb visible light. Different pigments absorb different wavelengths. Wavelengths that are not absorbed are reflected or transmitted, and that's why leaves appear green, because every wavelength is absorbed by the leaf except the green part, and that is what is reflected, and so the leaf looks green. And so this is just to review. This is the entire electromagnetic spectrum, and you go from the right to the left in intensity or wavelength size. So the biggest waves are the radio waves on the right, and the tiniest and most energetic waves are the gamma rays on the left. And somewhere in the middle there's this tiny little band of visible light, and that is what this is all about, this tiny little band of visible light. But if we look at the visible light, and this entire thing is actually just magnifying this tiny band, and what we see is that visible light is actually composed of a bunch of colors. And you start with the most energetic color, which is purple or violet, and you work your way all the way to the least energetic color, which is red, and that is 750 nanometers. So the shorter wavelength is on the left-hand side, and it has the higher energy. And the longer wavelengths have less energy, so the reds have less energy than the purple. And you know this from very early stages where you learned the visible light color spectrum, which was VIBGYR, violet, indigo, blue, green, yellow, orange, and red. So this is the same visible light broken up into its components shining through a chloroplast into the thylakoid membrane, where it is actually harvested. And except for the green part, which is reflected back, everything is used. What we'll see is that the main photosynthetic pigment is called chlorophyll A. But chlorophyll B is also present. That's an accessory pigment. It's slightly different in structure from chlorophyll A, but it broadens the spectrum used for photosynthesis. Other accessory pigments called carotenoids absorb the excessive light that would otherwise damage chlorophyll. And the carotenoids are the reddish colors that in fall, when the leaves are about to fall, the green disappears and the carotenoids are very apparent. This is what one chlorophyll molecule will actually look like, just in case you were interested in looking at it. Because how cool is this? You can actually see what a... Whoopsie. Touched the wrong thing here. And you can actually see what a chlorophyll molecule looks like. And here is the head of the chlorophyll molecule. It's a porphyrin ring. It's a light absorbing ring. So see, it's this pretty ring. It's a nice big ring. And in the center of the ring is a magnesium atom holding it all together. So this ring has a tail. And that is a hydrocarbon tail, just like we have in any other of our hydrocarbon molecules. And these react with the hydrophobic regions of proteins inside the thylakoid membranes. We're going to look at something else now. The photosystem. A photosystem consists of a reaction center protein complex surrounded by light harvesting complexes. And there are two types of photosystems, but let's talk about one first. So the light harvesting complex, those would be the pigment molecules bound to the proteins. They transfer the energy of photons to the reaction center. A primary electron acceptor in the reaction center accepts excited electrons and becomes reduced as a result. Solar power transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. Here we have the first step. Okay, so we have the photon coming in on the left, going into the light harvesting complex, which is all this purple junk, and then the center of that purple junk is the reaction center complex, which is all this blue junk, and then on the sides in the purple we see all these red dots bouncing around, and those are actually the that's the transfer of energy. And in the dead center, right dead center, what you see in the reaction center complex are a special pair of chlorophyll A molecules, these things are just pigment molecules, and the stroma is so the photosystem is this part, the light harvesting complex and the reaction center complex. So this and this, whereas the transfer of energy is happening here. So the photon is coming here, the energy is being transferred here into here. Okay, and then it goes from here. So the thylakoid space, which is in the interior of the thylakoid, is what is where the so again, this is the phospholipid bilayer. It's the thylakoid membrane this time. It's not a plasma membrane. And inside the plasma membrane is the photosystem, which is the part that actually harvests light or takes the energy from a photon of solar light and will transform it into what we want, which is chemical energy. There are two types of photosystems in the thylakoid membrane. PS2, which is photosystem 2, functions first because it's best at absorbing at a wavelength of 680 nanometers. So that would be close to the red part of this visible spectrum. The reaction center chlorophyll A of PS2 is called, therefore, P680. But there is another photosystem, PS1, that is best at absorbing at a wavelength of 700 nanometers. So that is even redder. And it's called P700 because that's just where it absorbs. During the light reactions, there are two possible routes for electron flow, cyclic or linear. The linear electron flow, which is a little bit more complicated than the cyclic electron flow, happens to be the primary pathway. And it involves both photosystems, 1 and 2, and it produces ATP and NADPH using light energy. In the cyclic electron flow, electrons cycle back from ferredoxin to the PS1 reaction center, but not PS2. All right, so let's look at the linear electron flow. There are 8 steps. In step 1, the photon will hit a pigment, and the energy is passed among the pigment molecules until it excites P680. An excited electron from P680 is transferred to the primary electron acceptor, and we can now put the traditional mark of a plus on it and call it P680 plus. So this is step 1. So the electron, the light is coming in. This is photosystem 2. It is bouncing around a pigment, and the electron is then sent to the primary electron acceptor. Step 2, what you have is in step 2, what we have is, well, actually, what we're looking at is water being split, and we are using that to go excite P680 plus. All right, so let's look at it. Water is split by enzymes. The electrons are transferred from the hydrogen atoms to the P680 plus, which is then reduced to P680. P680 plus is the strongest known biological oxidizing agent. How interesting is that? It is pretty darn powerful. Oxygen is released as a byproduct of this reaction. Each electron will now fall down an electron transport chain from the primary electron acceptor of PS2 to PS1. And energy is going to be released by the fall. And that will drive the creation of a proton gradient across the thylakoid membrane. The diffusion of the protons across the membrane will drive ATP synthesis, sort of like what we saw in the mitochondria. So here we are looking at the electron transport chain, same cytochrome complex business going on, and ATP is produced, but now it's going into the photosystem 1. We have the same idea, pigments accepting the electrons until the primary electron acceptor will take it, and then it will go down an electron transport chain to harvest all this energy, and we will produce a lot of energy. In PS1, like PS2, the transferred light energy excites P700, which loses an electron to an electron acceptor, and therefore becomes P700 plus. So that is the one that's missing an electron. It accepts the electron passed down from PS2 by the electron transport chain, and then each electron is going to fall down the electron transport chain from the primary electron acceptor of PS1 to the protein called ferredoxin. The electrons are then transferred to NADP plus, and then it is reduced to NADPH. The electrons of NADPH are available for the reactions of the Calvin cycle, and this will also nicely remove a proton from the stroma. Neat, isn't it? It all ties together. So here we have a summary of the linear electron flow. Instead of all those 8 steps, what we're looking at is, well, photosystem 2 will take water, give out oxygen, use a primary acceptor, and go down an electron transport chain in a cytochrome complex, go to photosystem 1, where the primary acceptor will then send it down another electron transport chain of ferredoxin, and you end up with NADPH. The mechanical analogy for the linear electron flow is sort of like that guy at the bottom of the left-hand side. He has this big giant hammer, which is the photon, and he's trying to raise a small ball, transfer the energy from that photon to that small ball and move it very high up, so it has potential energy. So he's hitting that seesaw. The ball rises because of the force, and the guy on top of a ladder, or photosystem 2, which is the first step of photosynthesis, will catch it. But then instead of tossing it to the guy on the next ladder, he's just going to slip it down this little chute, which is the transfer chain. 2 ATP are going to be formed, and then to amplify the signal, there's another guy with another great big hammer, which means another photon is used, and he's going to hit that electron really hard. So it's already not at ground level. It's got a bunch of energy still in it, and it will hit it so hard that it will go above to the second guy standing on the second taller ladder, who will catch it, but then he'll just kind of plop it into a little bucket and save it. In cyclic electron flow, electrons just cycle back from ferredoxin to the PS1 reaction center. So that's pretty easy. Cyclic electron flow uses only photosystem 1 and produces ATP, but not NADPH, and no oxygen is released. So that's just the downside. But it's pretty easy. So all we have here, we don't involve photosystem 2 at all. We're just involving photosystem 1, and so not much goes on. But some organisms, like purple sulfur bacteria, they have PS1, so they can exist. They just don't have PS2, so they can't actually do all of that. Cyclic electron flow is thought to have evolved before a linear electron flow, so it's an ancient form. Cyclic electron flow may protect cells from light-induced damage, so good thing. Chloroplasts and mitochondria. Let's see what are the similarities and the differences. Chloroplasts and mitochondria generate ATP by chemiosmosis, but they use different sources of energy. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into the chemical energy of ATP. So that's the reverse. In mitochondria, protons are pumped to the intermembrane space, and they drive ATP synthesis as they diffuse back into the mitochondrial matrix. In chloroplasts, protons are pumped into the thylakoid space, and they drive ATP synthesis as they diffuse back into the stroma. That's very similar. And so we have on the left-hand side a mitochondria, on the right-hand side we have a chloroplast. And what we're doing is we're showing that they are actually transporting electrons and producing energy. This is the current model of the thylakoid membrane. If you notice, it is a phospholipid bilayer. It is 2 membranes, so because it's a pancake, and there's thylakoid space, which has a high proton concentration, which is a good thing because it helps in driving the reactions. And then you have these photosystems embedded in the membrane, in the phospholipid bilayer membrane. So a photosystem 2, and then there's photosystem 1. There's cytochrome complexes, which will shuttle the stuff around, and you are going to end up with a bunch of ATP being produced. The Calvin cycle, like the Krebs cycle, regenerates the starting material after molecules enter and leave the cycle. The cycle builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. The Calvin cycle, well, what does it do? Carbon enters the Calvin cycle as carbon dioxide. It leaves as a sugar named glyceraldehyde 3-phosphate, or G3P. For net synthesis of 1 G3P, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide. The Calvin cycle has 3 phases. The first phase is called carbon fixation, and it is catalyzed by the enzyme rubisco. The second phase is called reduction, and the third phase is the regeneration of the carbon dioxide acceptor. Here we are, Calvin cycle. So we've already looked at the light reactions or photosynthesis, and now we're going to hook it up to the Calvin cycle to produce sugars, which is what we want. Briefly, the Calvin cycle is 3 carbon dioxides go in, and there's reduction. You get 1 G3P by reduction, and then there's regeneration of the carbon dioxide acceptor, and that would complete the cycle. Photorespiration, what does that mean? That is a new idea, but it's really important to understand it because it leads to some very interesting new ideas. Dehydration, which happens all the time on terrestrial places where the plants are sitting around, and they will lose a lot of water. So dehydration is a big problem, but it's going to require trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants will close their stomata, which will save water loss, but then it will also limit photosynthesis, which is unfortunate. So that's why in the desert, you don't get a whole bunch of flowers or plants growing all the time, because there's not a lot of water, and whatever water there is, it's so hot that the plant is not going to want to lose that little amount of water, so it won't even do a lot of photosynthesis, so it grows really, really slowly. So when the stomata close, they reduce access to carbon dioxide, and that causes oxygen to build up, and that is not what the leaf likes. And the leaf does not like oxygen to build up. It's just like when we don't like lactic acid to build up in our leg muscles after running. So these conditions will favor an apparently wasteful process called photorespiration. Only apparently wasteful, okay? It's not really wasteful. It's just apparently. There's 3 types of photosynthesis. C3, C4, and CAM. Most plants are called C3 plants. They initially fix carbon dioxide by rubisco and form a 3-carbon compound, which we know is 3-phosphoglycerate. But in photosynthesis, in photorespiration, rubisco adds oxygen molecule instead of carbon dioxide in the Calvin cycle. This is strange, right? So it produces a 2-carbon compound. Not good. Photorespiration will then consume oxygen and organic fuel and release carbon dioxide without producing ATP or sugar. That is why it is called wasteful. The C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into 4 carbon compounds. So there are 2 distinct types of cells in these plants, in the leaf of these plants. Bundle sheath cells, which are arranged in tightly packed sheaths around the veins of the leaf, or mesophyll cells, which are loosely packed between the bundle sheath and the leaf surface. So you have the best of both worlds. Sugar production in the C4 plants occurs in a 3-step process. So the production of the 4 carbon precursors is catalyzed by the enzyme PEP carboxylase in the mesophyll cells. PEP carboxylase has a higher affinity for carbon dioxide than rubisco does. It can fix carbon dioxide even when carbon dioxide concentrations are low. How cool is that? These 4 carbon compounds are exported to bundle sheath cells, and within the bundle sheath cells, they release carbon dioxide that is then used in the Calvin cycle. Here is a picture of a C4 leaf. What you see is a vein. Let me find my pen. Here we are. So here is the vein of the leaf, and around it you see the bundle sheath cells, and then which are tightly packed, and then around it are the mesophyll cells, which are loosely packed. You can see all these little holes, and then that's just the top of the leaf. So where is the C4 pathway? It is occurring in the mesophyll cells and this entire stretch. So right here, and this is just a blow up of it right here. So that is where the C4 pathway is occurring, and you have in the mesophyll cell, the carbon dioxide is going to become a 4-carbon compound, okay, not 3, and you go into the Calvin cycle and you get sugar. But anyway, the third type of plant is called the CAM plant, and some plants like succulents use crassulacean acid metabolism, or CAM, to fix carbon because they have a different problem. They only open their stomata at night. All right, and because it's too hot during the day, they can't open them at all. So they do need to open them because they need to breathe. So they will incorporate carbon dioxide into organic acids at night. There's no sunlight. Stomata will close during the day, and the carbon dioxide is released from the organic acids and used in the Calvin cycle. So they just take the carbon dioxide, they temporarily store it in organic acids, and then during the day, the stomata is going to close, and all those little organic acids are going to release the carbon dioxide to go into the Calvin cycle. Very neat, very tricky. So here we are with a nice summary slide. You have the roots of the plant taking up water, the leaves of the plant taking sunshine, and combining it inside the leaf, inside the thylakoid membranes. The light is harvested, sugar is exported and produced, and this is the review of photosynthesis, where the light reactions are on the left, and they're carried out by molecules in the thylakoid membranes. They convert light energy to chemical energy of ATP and NADPH, and they split water and release oxygen to the atmosphere. The Calvin cycle reactions take place in the stroma, not the thylakoid membranes. They use ATP, NADPH, to convert carbon dioxide to sugar, and they return ADP, inorganic phosphate, and NADP plus to the light reactions. So they just feed off into the light reactions. And that's it.